My name is Sam Keeping of Precision HEOR, and today I'll give an overview of the recently published manuscript entitled Comparative Efficacy of Cemiplimab for Advanced Cutaneous Squamous Cell Carcinoma, published in Future Oncology. Until the approval of cemiplimab, a programmed cell death protein 1 inhibitor by the US Food and Drug Administration in September 2018 and the European Medicines Agency in July 2019, there was no approved systemic therapy for patients with advanced cutaneous squamous cell carcinoma, known as CSCC. Cemiplimab has also been approved for use in Australia, Brazil, Canada, and Israel. This study systematically identified evidence on the efficacy of available systemic treatments historically used in patients with advanced CSCC. Additionally, the comparative efficacy of cemiplimab versus epidermal growth factor receptor inhibitors, otherwise known as EGFR inhibitors, pembrolizumab, and platinum-based chemotherapy was estimated using indirect treatment comparison methods, although trials had differing inclusion criteria and design. 27 systemic therapy studies were identified. Of these, 11 studies reporting overall survival, referred to here as OS, or progression-free survival, referred to as PFS, were included in the analyses. In this video, I would like to discuss the main takeaways from this study. The figures on this slide show the reported Kaplan-Meier curves for OS and PFS from the studies included in this analysis. Both outcomes were reported across all included studies except Picard et al., which did not report a Kaplan-Meier curve for PFS. Among all studies, median OS ranged from 11 months with dacomitinib and panitumumab to not reached with pembrolizumab in both CARSKIN and KEYNOTE-69. Median OS was not reached for cemiplimab in the phase 2 clinical trial based on the October 2018 data cut. The median follow-up duration for cemiplimab was 11.9 months. An estimated median PFS of 18.4 months was reported for cemiplimab. As can be seen in this figure, cemiplimab was superior to EGFR inhibitors in all indirect comparisons, with estimated hazard ratios ranging from 0.30 to 0.67 for PFS and from 0.07 to 0.47 for OS. Although it is not possible to draw conclusions from cross-trial comparisons, this figure demonstrates that the OS of cemiplimab was greater than that of platinum-based chemotherapy and pembrolizumab, with hazard ratios ranging from 0.17 to 0.41. This trend was also apparent in PFS when compared to pembrolizumab in the KEYNOTE-69 study, but not for other comparisons. In conclusion, our findings suggest that cemiplimab may offer improvements in survival relative to other systemic therapies in patients with advanced CSCC. As clinical evidence supporting cemiplimab matures and real-world evidence is collected, we anticipate that cemiplimab will continue to be a mainstay for treatment for patients with CSCC requiring systemic therapy.